Hi Hires, we are looking at metabolism and survival, and in particular key area 3, measuring metabolic rate. Okay, so first of all, let's look at what metabolic rate is. So we've already said that metabolism is the sum total of every single reaction that's taking place in the cell, catabolic and anabolic. So rate just means that I need to add in time. So metabolic rate is literally just the energy that's used over time. So that allows you to compare, because if you have a rate, then it's not just a random number. And that's really important when you're looking at trying to compare things at specific times or between different organisms. Okay, so here is really what we're looking at. This is how we get energy. So if we could measure any part of this, this would give us a rate. So food going in is not easy to measure really, because some of that might not actually be burned for energy. We could look at calorie intake, but not actually what exactly is being burned. Oxygen, that's reasonably easy. We could measure consumption over time. Carbon dioxide, we can also measure because you can do production over time. And energy as well, because energy we can look at just as heat. And heat we can measure quite easily because we can just take a temperature. So you should recognize not necessarily this specific diagram, but this idea of a food calorimeter from Nat 5. Okay, so the idea here was that in this little basin at the bottom we had dried powdered food, a weighed sample that we know exactly how much is there. And then we need some kind of ignition source, and in this case you would use a heated coil so that you don't have anything else being brought in. We have oxygen being pumped into this to make sure that everything burns, and then all of the heat that is released is absorbed by this water round the outside. Okay, and we've even got a wee stirrer to make sure that it's going up and down and keeping this liquid moving. And the exhaust gases that are being produced by the burning, they go through this copper pipe, which also transfers the heat to the water. So over every single bit that we can get out of this is transferred to the water, and the water we are taking the temperature with this thermometer over here. Now, because we know exactly how much energy is needed to heat up water, which is this. Oh, sorry. Let me just get that unit correct. Okay, 4.18 kilojoules per kilogram per degree C. Okay, so that means that you need 4.18 kilojoules of energy to raise 1 kilogram of water by 1 degree C. That's 1 litre of water. So if we know exactly how much this temperature has gone up and we know exactly what mass of water we have, then we can apply this formula, which you don't need to know for higher biology, but you'll have used it in physics and you'll use it in chemistry. So it's a really worthwhile one to see the link. Okay, so how are we going to actually measure this in living things gets a little, because we obviously don't want to just burn something. That's not going to be in a particularly good way to measure it. So we could use any form of calorimetry where we can actually just use this same idea, this conservation of energy. We cannot create the energy or destroy it, but we can change it in form. So we are looking for the heat produced. So we could put a human or any individual into a sealed container surrounded by water. That would not work out well for the human. Okay, so we have to use a slightly different system. So a human calorimeter is basically bigger. You use a container which is big enough for them to be in, like a little room, and inside that room you have incredibly well insulated walls, and then we use the water is connected inside with pipes, and the pipes are all hooked up with thermometers, and that's basically it. You can set different tasks, you can give different conditions, and you can look for the response in metabolic rate. There are easier ways to do this though. Okay, one of the easier ways to use the respirometer. So what we're looking for here is looking for the difference in that gas composition. So it's a lot simpler. Basically all you do is you hook someone up to a big bag and then you analyse the exhaled air. What you need is something that can measure carbon dioxide and oxygen, and then you're good to go. So there's lots and lots of different types of respirometers, and it does matter an awful lot what organism you're looking at and measuring. There are always some of the things that are always the same though. Okay, so we always have to get rid of the carbon dioxide from the air coming in. Okay, so we scrub the carbon dioxide out. So all you're getting in that is oxygen. Obviously not all, you're getting 20% oxygen roughly. You then have to remove the carbon dioxide from the exhaled air, and that gives you a difference, because remember, for every oxygen you make a carbon dioxide. So what we're saying is, if I remove the carbon dioxide at the end, that was how much oxygen was used, and now I have a difference in volume, and that lets me work out how much oxygen was there. This one you might have seen before, so just to remind you how this works, is a low-tech version of a respirometer. So we've got our little woodlouse sitting on a little platform in here, and then we've got our chemical that is removing the carbon dioxide. So in this whole section of air here, I'm just gonna, so all of this air and the air all the way up this pipe into here, okay, all of that air has no carbon dioxide in it. So every single time that this little woodlouse breathes in and out and then does respiration, okay, so it takes in oxygen, brings back out CO2, and that CO2 is just taken away straight away. So what actually happens is that it uses up a little bit of the volume of this yellow bit, all of the air that's here, and so it draws liquid up the tube, and that's how that works. Okay, so it's the same process, same idea. Get rid of the carbon dioxide to start with, and then get rid of anything that's produced, and that will reduce the volume because you've converted some of the oxygen to carbon dioxide. Right, so our next thing to consider is our basal metabolic rate. So this is your baseline. It's saying that that's the number of calories that your body burns at rest just to keep you ticking over. Doesn't matter what else you do in the day, this is just how much your body burns just to keep you going. And that it's not insignificant. You have lots of active transport that needs to run, you have things that need to happen inside your body, like your heart needs to pump, you need energy for that. So everything that just keeps your body going. Okay, but that doesn't stay the same throughout your life. It's going to change with all sorts of just basic things about you, and very much about the diet that you have and the exercise habits that you have. Okay, so to bring all that together, the metabolic rate in species is going to range hugely. Some species are going to have a very high metabolic rate, some are going to be very low. And depending on that demand, that's going to make a big difference about how they get that energy, which is what we're going to look at a little bit further on when we're looking at heart structures. So your niche that they have is going to decide how much energy you need to use to produce, and that's going to make a big difference to what's going on. And here's a little graph which I think is quite nice, showing you the mass of organisms and then also their specific metabolic rate. And you'll notice that actually you might consider that big animals would have the biggest rate, but actually that's not the case at all. They have a much lower rate than some of the smaller animals. So just give some thought to why that might be the case, because we're going to look at that a little bit later on. Okay.